In this video, I'm going to talk about the catalase test. So catalase is an enzyme that's released by microorganisms in order to help protect themselves against hydrogen peroxide. Hydrogen peroxide is a waste product. It is the result of cellular respiration. So when we talk about cellular respiration, we go through glycolysis, the Krebs cycle, the electron transport chain, and if this is aerobic respiration, we're going to use oxygen as our terminal electron acceptor. So the hydrogens are going to combine with electrons, and that oxygen is going to accept those electrons to produce water. Sometimes instead of H2O, H2O2 is produced, and H2O2 is hydrogen peroxide, and that is harmful. So to test for the presence of this enzyme, we do a simple transfer of bacteria to a microscope slide, and then we're going to evaluate that microscope slide for the presence of the enzyme by adding hydrogen peroxide to it. So we start by sterilizing the loop, and then we're going to take a small amount of bacteria and add it to the microscope slide. So it's just a dry microscope slide, and we're going to smear some bacteria around. Now I'm actually going to do two different species next to each other so that you can see two results. So now I'm going to add hydrogen peroxide. Hydrogen peroxide, it's just 3% hydrogen peroxide. This is the same grade that you can buy over the counter at any store. And I'm going to add a drop to the bacteria directly. You can see the bubbles. Now I'll bring this a little bit closer so you can see. Instantly the bubbles are forming. That one is catalase positive. And you can see bubble formation on this one as well, and that one is also catalase positive. Catalase positive means that the microorganism produces catalase. When catalase is present, it's going to break down that hydrogen peroxide to release gas, and that's why you're seeing the bubbles start to form. It's because gas is being released when hydrogen peroxide is digested by that catalase enzyme. This gives you immediate results, and you can record those results as positive for both of these samples.